Awesome. All right. So good to be here. I'm Joe Steiga, currently at Mount St. Joseph University in Cincinnati, Ohio. This is some work that I did a couple years ago with Ryan Early at the University of Alabama, focusing in on morphological integration and development in a pretty awesome species of fish, I think, the mangrove rivulus fish. So first, just to give you some context, the Paleozoic water to land transition was a major event in the evolutionary history of vertebrates. Early transitional species or fishopods faced numerous challenges as they began to exploit resources at the intersection between water and land. One of the big stressors was an increased gravitational load. This was a major hurdle to early transitional vertebrates, slowed down their stride, required a higher energy investment to facilitate effective locomotion. Stress posed by increased gravitational load likely contributed to several important adaptations, including the development of denser fused vertebral elements, which helped to facilitate stiffer joints, allowed for more support of their bodies to overcome gravitational load, while also protecting elements within the vertebral column itself, the spinal cord and various blood vessels during bending. Studying these adaptations and the mechanisms driving their evolution is made difficult by the scarcity of representatives in the fossil record. As such, modern day transitional species may help fill in these gaps. So big one historically has been mud skippers, which are shown over to the right here. This is work from Tran et al. In earlier this year. So understanding the full scope of phenotypic responses to development, to selection, understanding the historical ramifications of selection requires a multivariate perspective, given that traits are often integrated. Strong correlations among morphological traits, what is termed morphological integration, may highlight historical selection on performance. Selection on performance often comes with morphological consequences, changes. Of course, these constraints, this non-dependence among traits can facilitate evolutionary change, can bias evolutionary trajectories. Thus, it's important to not only understand correlations among traits to highlight historical or potential historical ramifications of selection, but also there's a predictive component here. These help to explain lines of least evolutionary resistance, and of course, correlations can change across development. So what did we do? We assessed morphological integration, phenotypic variants, genetic variants, phenotypic correlations, genetic correlations in the mangrove rivulus fish. We looked at over 1,000 individuals from across 50 plus genotypes, and we focused in on 3 different stages of development, or 3 different time points in early larval development. We looked at hatchlings at 1 day post-hatching, 15-day post-hatching individuals, 100-day post-hatching individuals. All of these individuals were grown in a common garden. And again, we're really interested in integration here. We think this is a good model to understand those factors or those mechanisms associated with morphological evolution in early transitional vertebrates, because the fish spends a significant time out of water on land, where it is an expert at terrestrial locomotion. So as you see over to the left here, courtesy of Ben Perlman, we see an individual actually engaging in a tail flip jump. So what did we do? We looked at 1,000 individuals. Again, we measured 5 different traits: standard length, We looked at the length of the neural spines, which are these splint-like structures on top on the dorsal side of the vertebral column. We looked at the length of the hemal spines in the posterior 12 vertebral elements. We looked at the length and the height of these elements as well in cleared and stained individuals. We hypothesized significant integration at each of these developmental time points in these vertebral column traits. Further, we expected morphological integration to be significantly stronger in the older individuals at 15 and 100 days post-hatching relative to 1 day post-hatching. Considering that these older individuals are likely to experience greater gravitational loads. They tend to spend more time out of water, as far as we know. So what we did statistically, we built univariate and multivariate mixed models in ASREML to produce phenotypic and genetic variance covariance matrices. And I'll talk about these more in a second. For these models, we entered standard length, this variable right here, as a fixed effect. To reduce the dimensionality of data, we conducted a PCA. We focused in on principal component 1 because this, for each of the traits we looked at, this explained about 94+% of the variation within these traits. To assess significance, we produced parametric, or using parametric bootstrapping, we produced 95% confidence intervals for each of these traits. So just to kind of orient you to a P matrix and a G matrix, what these things look like here, we have traits on the vertical axis. We have the same traits on the horizontal axis here. Along the diagonal, we have variance components. So for the P matrix, this is among individual phenotypic variance. For the G matrix, this is going to be among genotype phenotypic variance. That's what these measures are here along the diagonal. In the off diagonal, we have the correlations, either phenotypic correlations, which we're going to see in the P matrix, or among genotype correlations we're going to see in the G matrix. We also looked at matrix trace. This is the sum of the variance components. These are the sum of all the values on the diagonal, and reflects the total amount of phenotypic variation among individuals, the variation in the total multivariate phenotype. Our measure of integration was mean squared correlation. This is the sum of the squares, the correlations squared, each one squared, and then summed together, divided by the total number of correlations. It helps us to understand the phenotypic integration across the multivariate phenotype. So let's jump right into the results. I'll orient you to this table real quick before I dive into it. Each age group, 1 day, 15, and 100 day post-hatching. For each one of these age groups, we estimated a P matrix and a G matrix. The first line is matrix trace. So this is variance across all traits. The second line is the mean squared correlation, our measure of integration. Below the estimate, we have 95% confidence intervals. The confidence interval doesn't include 0, then it's a significant result. So pretty much what we see here is that yes, our traits we see a significant amount of phenotypic and genetic variance for our traits at each age group. There's a significant amount of integration, phenotypic and genetic, for each age group. When we compare the matrices, and this is where I think we start to get into something pretty interesting, when we're looking at the difference between the matrix we estimated at day 1 versus day 15, we look at the matrices between 1 and day 100, and then 15 versus 100. What immediately becomes apparent is that we see variance, both phenotypic and genetic variance, increase with age from 1 to 15. Or I should say, we see a significant increase between 1 and 15 and 1 and 100. Same thing between 15 and 100. So variance is increasing. Integration is also increasing between 1 and 15 and 1 and 100, although that is not always significant. We did not see a significant difference in integration between 15 and 100. It's kind of interesting. So when the difference is negative, it's telling us that either trace or mean squared correlation is larger in the latter age group. So 15's integrations larger in 15 relative to 1, larger in 100 relative to 1. So I thought this was pretty interesting. I think what this speaks to is really the difference in how these different age groups utilize their environment. Again, day one individuals are much more likely to spend a significant time in water. And we think this is the case because of a number of reasons, mainly surface area to volume ratio. Those individuals would be more likely to desiccate in terrestrial environments if they don't find nice damp leaf litter. Whereas 15 and 100, maybe that's not as big of an issue. In addition, 15 and 100 day individuals are better jumpers. They're more able to locomote effectively on land. So really, I wanted to dive a little bit deeper. What I'm showing you here, it's a ton of stuff. Sorry about that, but these are the different matrices for each age group. So here's 1 day post-hatching, the P matrix, 15 day, 100. And then on this side, we have the G matrices, 1, 15, and 100. So for the most part, what we see for these traits, and we see significant positive correlations, focusing on those right now at 15 and 100. But we see something pretty interesting, I think, at 1 day post-hatching. There's a couple things that I'm highlighting and I'll dive into. But really what I want you to take away from this slide is that it seems to be something is happening at 1 day post-hatching relative to the other two age groups. And we're going to dive into this in a second. So again, one of the things that we saw at 1 day post-hatching was the correlation between vertebral element height. So it's going to be the height of the vertebral element or the height of the vertebral centrum here. And then hemospine length, the length of this structure, how long this structure actually is. We saw a negative correlation between these two traits, a potential trade-off between these two traits. We saw that in the P matrix, and we also saw it in the G matrix. So what I'm doing here over to the right, I'm kind of giving you a kind of simplified version of what this might reflect. So at the one end of this gradient, we see a combination of traits where the vertebral elements are tall, the hemospines are short. On the other side of this in pink, we see the opposite trends. The vertebral elements are relatively short, hemospines are long. This might reflect a trade-off between vertebral column rigidity, what we would associate more with this morphology. Down here, maybe more associated with flexibility, or I should say, the ability to store more strain energy in the joints. When these hemospines are going across several joints, intervertebral joints, this morphology likely stores more strain energy, which may be important if you're a tiny fish trying to escape a predator. So this is the bottom part of the vertebral column, the vertebral elements in the hemospines.
Great. Sorry, Joseph, I'll just ask you to wrap up.
Okay, yeah.
It's 14 minutes now.
Yeah, so other side of this, what we see on the neural spines was kind of interesting. We saw the same trend in the G matrix, but really we saw an opposite trend in the P matrix, which I thought was interesting. And this may reflect phenotypic plasticity in response to some uncontrolled environmental variable in the common garden. The main takeaways, we saw a strong integration in the later age groups, 15 and 100. Something particularly interesting appears to be happening at 1 day post-hatching. like to thank my colleagues, helped me out with this: Alistair Wilson, Tom Housley, Ben Greenberg, Andrew Burks. Thank you for listening.
Great. Thank you so much, Joseph.
Yep.
Appreciate that. So in the interest of time, we'll move on to our next speaker. If you have questions for Joseph, you can send him direct messages in the chat. So Diana, whenever you're ready.
All right. Hello, everyone. Thanks for joining today. My name is Diana Renison, and I'm an assistant professor at UC San Diego. Oops. So when we go out to nature, we can find repeatability of evolutionary patterns at a variety of different levels, and these can be referred to as parallel or convergent patterns of evolution. I'll either use parallelism or repeatability interchangeably throughout the talk. But a fantastic case of repeatability is seen in the Caribbean anoles, where we see entire ecotypes repeatedly evolving across independent island replicates. In a variety of systems, we also see this repeatability or parallelism at the level of phenotypes, even among distantly related taxa adapting to similar ecological conditions. And in many cases, these phenotypic similarities are underlined by the use of the same genes or even genotypes. So we can find repeatability emerging at a variety of scales. And with all of the whole genome data coming online, we can also investigate these patterns at a genome-wide level, looking at repeatability of patterns of genomic divergence. So that's led to some new questions or, core questions in evolutionary biology. How does parallelism or repeatability differ across biological scales, from phenotype to genotype to genomic and even ecotype? And do these estimates between phenotypes in particular and these genetic estimates scale in a predictable manner? So I'm going to address that question using the model system of three-spine stickleback. I'm using two different ecotype comparisons. So the first one is benthic and limnetic stickleback. These sympatric species pairs or ecotype pairs have evolved multiple times in lakes in British Columbia, Canada. So they exhibit ecological divergence, which is underlined by some degree of phenotypic parallelism. So here are three of the extant replicates, and hopefully you can appreciate that the bigger one, which is the benthic, looks more similar in its form and overall shape across lakes than it does to its sympatric limnetic form, which is more streamlined, smaller repeatedly across lakes. And there's a variety of different behaviors, ecologies, and morphologies that have evolved in parallel. In stickleback, we also have these stream and lake ecotype pairs. These have evolved thousands of times throughout the Northern Hemisphere, and they're found in parapatry within watersheds. They are thought to exhibit more variable phenotypic parallelism. You can even just see in this figure here that the similarities in their morphology is less consistent even between just these two watersheds. So there's some aspects of shape that are kind of repeatable between streams, but these lake fish actually look quite different from one another. And there's also thought to be some ecological similarity and parallelism between these ecotype pairs in terms of the pattern of divergence. So stream and lake pairs and benthic and limnetic pairs are thought to be kind of diverging along the same axes. So stream individuals tend to have this deeper bodied morphology and exploit benthic resources, whereas solitary lake fish tend to be more zooplanktivorous like the limnetics. So I'm going to do some different comparisons both within and between these different ecotypes. So the approach we used to look at this question of repeatability and scaling was that we surveyed the 3 extant benthic limnetic pairs and 4 of the stream lake pairs. We stained specimens with Alzarian red, which binds to calcium and helps to highlight bony elements. So these are an example of some specimens. And then on these stained specimens, we phenotyped 12 ecologically relevant traits, largely those involved in defense against predation or trophic resource acquisition. In terms of the genetic approach, we did genotyping by sequencing for 20 individuals per population, so 20 benthics, 20 limnetics from all of the replicates and so on for stream and lake. We estimated genetic divergence using FST and DXY for more than 35,000 SNPs. And then to quantify parallelism, traditionally, both in this system and a variety of other systems, people have used kind of a bivariate approach saying, yes, divergence is in the same direction for a given trait or a particular candidate gene, or no, it's not. I wanted a more quantitative, holistic measure, so I employed more recent multidimensional vector analysis methods. So what you do basically is for each population, you estimate the data in multi-dimensional space in using either the phenotypic or genotypic data. And then you're able to look at how much divergence there's been between paired populations. So for example, this would be Paxton benthic against the Paxton limnetic. You can estimate how much divergence there's been between the pair. And when you have replicates, in this multidimensional space, you can look at how similar those trajectories are. So for example, this would be the benthics and limnetics from Priest Lake. And when you have an angle between these vectors that's very small, let's say 0, that means that they're diverging in exactly the same manner. When you have an angle that's close to 90, that's the null expectation, and there's not really any evidence of repeatable or parallel evolution. And when you have angles greater than 90, it's indicative of anti-parallelism. So in fact, the pattern is divergent between the pairs. So we did this analysis for both phenotypes and genotypes, and we also did it for benthic limnetic comparisons like is depicted in this example, but also stream lake pairs. So you'd have a stream population and a lake and another replicate stream and lake comparison and estimate that angle. And then we also did a between ecotype comparison where we looked at benthic limnetics to stream lakes to look at how similar those evolutionary trajectories were as well. So jumping right into the results. So first looking at phenotypic parallelism. So you can see here, hopefully, we have these benthic limnetic comparisons, stream lake comparisons, and then benthic limnetic to stream lake, the between ecotype comparisons. And on the y-axis, we have the parallelism estimate, which is theta, that angle between those vectors in multidimensional space, this time based on phenotypic data. And the dotted line indicates the null expectation of 90 degrees. So you can see that for the benthics limnetics, the angles tend to be rather small. They're among the smallest estimates in any comparison. So there's quite some evidence of phenotypic parallelism among these replicates. We see more variable and less magnitude of parallelism in our stream lake pairs. But interestingly, we do find some evidence of phenotypic parallelism between the stream lake and benthic limnetic comparisons, supporting this idea that they're adapting to kind of similar niches. When we looked at the genomic data, we have, again, these 3 sets of comparisons. We have the parallelism estimate, again, theta on the y-axis. And you can see that for the benthics and limnetics, again, there's quite small angles, much smaller than 90 degrees, and they're consistent among all of our replicates. For the stream lake comparisons, they're smaller than 90 degrees, but much greater than the benthics limnetics, indicating that there's less repeatability in the genome-wide patterns of genetic divergence. And then what I found was really interesting was that between the stream lake and the benthic limnetic pairs, there's no evidence of parallelism. In fact, they're using completely different regions of the genome to generate that phenotypic parallelism that we found evidence of on the prior slide. So that was pretty cool. Getting to the kind of broader picture of the scaling of phenotypic and genotypic or genomic parallelism. So we see greater parallelism, so smaller angles generally for phenotypes relative to genotypes. So you can see on this plot here, our phenotypic estimates on average are smaller than our genotypic estimates with some outliers. And these outliers are the estimates from the benthic limnetic pairs. So we actually find quite some repeatability in those patterns, but it's kind of atypical of what we're seeing in other comparisons. In terms of the predictability, the scaling between phenotypic and genotypic parallelism within any given comparison, we find no significant relationship. So the regression line here is across all of the 3 different ecotype comparisons. But the colors indicate within, and in no case is there kind of a significant relationship between the phenotypic parallelism estimates and the genotypic parallelism estimates. So it's really difficult to predict kind of what level you'll see, because you can find a high level of phenotypic parallelism and the same degree of genomic parallelism as when you find a lower estimate of phenotypic parallelism. So for stickleback aficionados, kind of the system-specific summary is that sympatric pairs of benthic limnetics exhibit disproportionately high degrees of genomic parallelism. The phenotypic divergence between benthic limnetic and stream-like pairs actually is underlied by some similarity of these evolutionary trajectories. And interestingly, that pattern is generated through fundamentally distinct genetic mechanisms. So they're not using the same genes or alleles to generate those similar phenotypes. The take home on more general evolutionary processes is that parallelism tends to be greater for phenotypes than genome-wide genotypes, but the scaling of that relationship is unpredictable. So I think the next steps are really trying to dig into the ecological and demographic processes that are generating this variation that we see among replicates. Are there certain factors that we can use to try and understand why we find variation in the estimates of phenotype and genotype? So looking at similarity of the selective landscape and estimating founding population and things like that. So several people to thank. First and foremost, an undergraduate in the lab, Denise Meyer, who did all of the phenotyping for the project. The genotypic data was collected in collaboration with Yoel Stewart, Dan Bolnick, Dolph Schluter, and Katie Peichel from a variety of prior projects, for which I was funded by the Marie Curie Fellowship, and I'm currently funded by the University of California, San Diego. And I'll just leave this up. I have some new funding in the lab and some PhD student and postdoc opportunities. So feel free to send me an email if you're interested in any of those things. Thank you for listening.
Great. Thank you so much, Diana, for a wonderful talk. We have a couple minutes for a question or two, if anybody would like to ask. Ideally, using the hand up. Oh, you have some hands.
Okay. Hopefully I'm not butchering too badly. Go ahead.
Sorry, who are you calling on? We have to ask them to unmute. So
Narcissus.
Oh, sorry.
Hi, Diana. Do you hear me?
Hi. Yes. Mm-hmm.
Yeah, perfect. Because I used two computers. Thank you very much for this interesting talk. My question is, maybe I missed it. How did you look at the genome? Did you look at the only genome-wide patterns, or did you look for outliers, or how did you want to relate the phenotypic and genotypic parallelism?
Yeah, so for this talk, I was just showing you the whole genome-wide pattern, which is summarized by basically looking in multidimensional space at the similarity of allele frequencies among the replicate pairs, the similarities of the difference in allele frequencies between benthics and limnetics among the replicate pairs. But we've also done outlier analyses to look at how similar specific regions are, and we find that there's quite some parallelism for specific loci in the benthic limnetics, not much repeatability in the stream lakes, and then they fundamentally do not use the same alleles between the benthic limnetic and stream lake comparisons. Like, they're never overlapping. There are no overlapping candidates of outliers between the benthic limnetic and stream lake pairs. So yeah, we've looked at both, but I didn't have time to kind of dig into everything today.
Okay, thank you very much. There are other people to have questions. Maybe I write you more.
Sure, great.
Okay.
Rex?
I'm gonna interrupt. I'm sorry. I think we do have to move on to the next speaker. But if people have more questions for Diana, you can send direct messages in the chat. So, sorry to cut you off. Javed, you're up.
Right, can you hear me?
Yes.
Right, hello everyone. My name is Javad and I'm a master's student in Sally Alto's lab in Biodiversity Research Center at UBC. And today I'm gonna tell you about my research about predicting fitness in different environments using some fundamental trade-offs of growth. And basically, I'm gonna talk about costs of adaptation. So first of all, I'm gonna tell you about what led me here. So in our lab, there is this big experiment going on where my lab mates are adapting these to lethal concentrations of different heavy metals. And when they, for example, adapt them to copper, then they test the fitness of copper adapted lines to in all different heavy metals. And they are interested in these pleiotrophic effects. So they had one line that was able to grow pretty fast in all metals, and it had only one mutation. And that mutation was in glycolysis, which is the pathway that cells use to eat food. And that made me think that is it possible that that cell is basically just eating food faster, and it has more energy, therefore it can deal with all the metals. And I know that metabolism is much more complex than that, but that was my initial reaction to that. So I thought about it more. I found this question really interesting. I wanted to know when is it beneficial for organisms to have adaptation to stress tolerance, and when it's not, when is it just better to keep eating food and growing as fast as they can and just tolerate some death rate. And also I was interested in how environment quality, for example, I don't know, food quality or energy availability affect the cost of adaptation. And when I say cost of adaptations, I'm thinking about a scenario where cells are growing and the stressor is causing some death rate, and the adaptation can prevent that death rate, and the cost of adaptation is growing slower. So the cost of adaptation is an absolute reduction in birth rate, and benefit of it is reduction in death rate. So at first, it seemed to me that, okay, probably organisms that have access to more energy can afford having adaptations easier, and they probably pay a lower cost. Also, it seemed intuitive to me that faster growing organisms probably would pay a higher cost, and their growth rate would decrease faster, because slow growing organisms it's not possible for them to have a huge reduction in growth rate. And these two intuitions were kind of contradictory, because to me it seemed like that, oh, probably faster growing organisms also have access to more energy, but not completely. So I was really puzzled. So I needed a framework that allows me to think about growth rate of cells in a much more clear way. And I started reading, and I encountered something that was really interesting for me, which is bacterial growth law. So since 7 years ago, people have been observing that as they grow cells in foods with higher quality, and by higher quality, I mean a better sugar, like simple sugar, like glucose, the growth rate of cell increases, and also fraction of ribosomes in the proteome increases. And they couldn't measure fraction of ribosomes. They could measure the ratio of RNAs and proteins in the cell, which is a proxy for fraction of ribosomes. And so they were observing this robust linear positive relationship between growth rate and fraction of ribosomes when altering nutrient quality. And also they were observing that when they reduce the efficiency of ribosomes by adding some antibiotic that inhibits ribosomes like chloramphenicol, there is a linear negative relationship between growth rate and fraction of ribosomes. So the way that they explained these results was by coming up with a model. Oh, also they observed that the fraction of ribosomes never goes above a certain threshold. So they explained it using this 3-compartment model of proteome. They were like, in a cell, okay, if probably a fraction of proteins is allocated to a structure and maintenance. And so that fraction is fixed, cell has to allocate those for structure, those proteins. And then the rest of proteome is allocated between ribosomes and related proteins, and also metabolic proteins that provide substrates for ribosomes. And when cell is in, so they assumed that they assume that cell is always has enough enzymes that provide substrate for ribosomes to saturate ribosomes. And they were like, okay, when cell is in a high quality food, it's an easy sugar, cell can saturate ribosomes and provide enough substrate for them by allocating a little bit to proteins that do processing. And there is more room left for ribosomes, so cell can allocate more to ribosomes, and ribosomes do protein synthesis, and cell is mostly protein. So basically they are doing biomass synthesis and cell growth. And so cell grows faster, and also it has a higher fraction of ribosome. Also, when they inhibit ribosomes by antibiotics, cell grows slower, but increases its fraction ribosomes to make up for some of that loss. And that's why they are observing these relationships. And notice that I'm gonna use fraction phi r for fraction ribosomes, phi p for fraction of enzymes that are allocated to metabolic processing, and phi q for structure and maintenance. And so they explained this model, but they knew that their assumption is not correct and ribosomes are not always saturated. And I was like, okay, maybe I can model the trade-off between ribosomes and other enzymes. And if I do that, I have a framework to think about growth rate. So when cell grows, when a cell doubles, everything in a cell doubles, right? So the birth rate of cells is equal to birth rate of ribosomes. And notice that I'm talking about birth rate, not growth rate, because I'm thinking about a simple scenario where cells are just growing and there is no death rate, just for simplicity. So birth rate of cells is equal to birth rate of ribosomes. And birth rate of ribosomes depends on how many new ribosomes each ribosome makes in unit of time. And that birth rate is equal to how many total proteins the ribosome makes in a unit of time and what fraction of them are ribosomes. So as you can see here, ribosome makes both new ribosomes, both new ribosomes, new metabolic proteins, and new maintenance proteins. And so maintenance proteins are there, but cell can change the ratio between fraction R and fraction P. But there is a trade-off here. If cell increases fraction R, there will be more new ribosomes and higher fraction of new proteins are ribosomes, which means higher birth rate of ribosomes. But there are not enough enzymes to supply ribosomes, so the translation rate goes down. But if cell wants to maintain high translation rate, it needs to reduce fraction of ribosomes. So there is a trade-off there, and there should be an optimum fraction of ribosomes. So I look at this graph. Focus on the blue line. The blue line is translation rate per ribosome, which is gamma as a function of phi r. Phi r is fraction of ribosome. So x-axis in this graph is fraction of ribosome. As I said, fraction of ribosomes can't go above a certain threshold because there are some enzymes allocated to structure, some proteins allocated to structure. So when fraction of ribosomes is very high and there is no metabolic protein, translation rate approaches 0. And when it's very low and there are many metabolic proteins, ribosomes will be saturated and translation rate, which is the blue line, approaches its maximum. And birth rate of ribosomes is equal to translation rate, which is the blue line, times fraction ribosomes. Fraction ribosomes is the green line. It's a 1-to-1 line as a function of fraction ribosomes because x-axis is also fraction ribosomes. And birth rate of new cells is the orange line, which is the result of multiplication of green and blue line. And as you can see, there is a fraction of ribosome that maximizes birth rate. And when cell is in that maximum, it's allocating that much to each of those fractions. So remember that at first I was concerned about how quality of food changes the growth rate of cell. And I can incorporate that into that framework by the slope of this line. So the slope of this line depends on food quality. When food quality is high, the slope is higher or actually more negative, because a little bit of proteins in fraction P can provide a lot of resources for ribosome and like saturate them faster. And when food quality is low, the slope will be less negative. And I was like, okay, perfect. This is the framework that I have. And if you are interested about how I formulated the blue line mathematically, I just assume that translation rate is a hyperbolic function of number of metabolic enzymes per each ribosome. So this is the simplest form that I could think of, but possibly can have other forms. But we don't care about that for now. We care about the graph, and we want to know whether this can predict, this can recreate those bacterial growth laws. And so I tried, and I was like, okay, perfect. It's doing what I expected it to do, and I can use this framework to think about bacteria, like recreate those results of like when I change nutrient quality, there is a positive relationship between growth rate and fraction R, and when I do translation inhibition, there is a negative relationship between fraction R and growth rate. I was like, perfect. So now I want to use this framework to think about costs of adaptations. Okay, look at this graph and focus on the blue line. The y-axis is growth rate or birth rate actually, and x-axis is fraction Q, size of the fraction that is allocated to structure and maintenance, and not growth. So when a cell is not in that blue line, look at the blue line. So when fraction Q is very small, the growth rate is high. And as size of fraction Q increases and it reaches 1, the growth rate goes to 0, because when fraction Q is 1, there is no protein allocated to fraction P or fraction R. And those two fractions are in charge of growth. And the linear form of this function is because of the way that I have formulated the model, and it's consistent with some experiments, but that's not necessary for my conclusion. So, and when a cell is in low quality food, the orange line, it starts somewhere lower in y-axis, but it goes to 0 at the same point, at almost the same point, right? So the growth rate based on this model depends on size of fraction that is not allocated to maintenance, which is 1 minus fraction Q, times the slope of this line, which I like to call return on investment, because it tells us when a cell allocates part of its structure to growth, how much it grows. And cost of adaptation depends on how much an adaptation changed fraction Q times the slope. Because I don't know, for example, pumps or chelators that help cell deal with heavy metals, they are not participating in growth, right? They are just taking away from the fraction that was available for growth. So now let's think about cost of adaptations. Oh, and before that, I just wanted to show you that people have been doing some experiments in E. coli, and when they express an unnecessary protein for growth, there is that reduction, linear reduction in growth rate. Okay. Now, look at organism A and B. The A is in high quality environment, B is in low quality environment, and both of them are allocating the same fraction to maintenance. And they can have an adaptation that requires higher allocation to maintenance, and it reduces birth rate, right? And so cost of that adaptation, which is the reduction in growth rate, will be lower when food quality is low, right? Because the slope is lower. You might think that what I'm basically saying is that organisms that are already growing slower pay a lower cost, which is one of the initial intuitions that I have, but that's not what I'm saying. It's all about return on investment. Here, look at organism A, B, and C. C is growing the slowest, but A and C are both in high quality food, and B is in low quality food. C is growing slow because it's already allocating a lot on maintenance.
Javid, I'll just ask you to wrap up. We're past 14 minutes now.
Sure. And so the cost of adaptations, as you can see, only depends on the slope of the line, and it has nothing to do with growth rate. And what about adaptations that require energy? That also can be translated to structure allocation, because we can say what fraction of a structure could be allocated to provide that energy. So what I'm predicting is that whenever return on investment is low, organisms tend to invest more on stress tolerance. And those adaptations tend to be involve more structure allocation when return on investment is low, which is basically when food quality is low. And notice that whatever I said is not anything particular about cells. It can be applied to other organisms. At the end, I would like to thank my lab, thank Sally for being an amazing supervisor, and thank Penny for her experiment that inspired me, and Ali that helped me to think simple and keep my models as simple as possible. And thank you all for your attention.
Great, thank you so much, Javid. And we will move on to our next speaker. As before, if you have questions for Javid, please send private messages in the chat. And Shuba.
Is my screen visible?
Your screen is visible. Yeah, great.
Hello, everyone. I'm Shubha, and today I'm going to speak about some of the work that I've been doing on understanding the impact of founder context on adaptation. Under environmental changes, the adaptive abilities of organisms are of paramount importance. Among the various factors that affect adaptation, the founding population size and standing genetic variation are vital to population adaptation. However, in these situations, does the identity of the founders or their experience matter for adaptation? Studies in the recent past have shown that individual context or experience actually matters. For instance, in the mealworm beetle, Tenebrae molitor, adult density and age affected their fecundity. And in Wendy's fish, young females produced more and smaller eggs than older females. This is just a small representation of a large body of literature that suggests that individual context or experience impacts their fitness and behavior. This is also something that we saw in the flower beetle, Tribolium castaneum, which is a generalist pest. This is work that was carried out by Brinda, who was a graduate student in our lab. When you take females that are either young or old, that have experienced either low or high levels of competition, and present them with an optimal resource, which is wheat flour, a suboptimal resource, which is finger millet flour, where development is slower, not only do these females lay different numbers of eggs, but they also allocate their eggs to the suboptimal resource differently. As you can see, young females that experience low density lay the highest number of eggs, and compared to that, the young females that experienced high density lay fewer eggs and also allocate a smaller percentage of these eggs to the suboptimal resource. This is something that we see in the short term. Is it likely that these effects can percolate over generations? In short, does founder context influence colonization success? To address this question, I initiated populations that were founded by females which were either young or old, or that experienced low or high density in a full factorial combination. Colonization success can be looked at from the point of view of population demography. I carried out oviposition assays on the females from the different contexts, similar to what you saw on the previous slide. And based on those observations, I expect that population sizes will be larger when the females have a higher fecundity. Another aspect is egg allocation to a new resource, and also niche expansion, which is the consumption of a new resource. For the purpose of this talk, I will focus on population demography and egg allocation to a new resource. To start with, we need to obtain founders of different age and density contexts from the laboratory stocks. For young individuals that experienced low density, I isolated the individuals as pupae and used the adults when they were 2 weeks. Similarly, for old individuals that experienced low density, I isolated them as pupae, but this time allowed the individuals to age in isolation post-mating till the age of 6 weeks. For young individuals that experienced high density, I took the adults from stock populations at 2 weeks of age. And for old individuals that experienced high density, I took the adults from stocks when they were 6 weeks old. There are 2 founder age and 2 founder density contexts, creating a full factorial combination of 4 treatments. And I had 6 replicate populations in every treatment. I took 100 adults and allowed them to lay eggs in a habitat that contained both wheat and finger millet flour in adjacent patches. As you can see in this photograph here, there was no barrier between the resource patches. After 6 weeks, I count the number of adults and pupae in the populations and allow 100 adults to lay eggs in fresh resource to start the next generation. The remaining individuals were frozen for chemical and physiological analysis. I used the census data as a measure of population size. There are 2 important points here. First, the contexts that you will hear of represent that of the founding individuals and not the individuals of subsequent generations. 2, every generation is initiated by 100 adults. On the left, you can see how the habitat looks when the adults have started laying eggs in them. And to the right is how the habitat looks close to the census time point. And I mark on the box the boundary between the 2 regions. Over the course of experimental evolution, I conduct periodic oviposition preference assays of the females from these populations. I measure oviposition preference by placing the female with the 2 resource patches. And after 48 hours, I count the number of eggs she has laid in each patch. Let us see what happens to fecundity between generation 1 and 6. As you can see from these plots, in the young LD founder context, these are populations founded by young females that experienced low density. There was a decline in fecundity from generation 1 to 6. But in all the other founder contexts, there was an increase in fecundity. At generation 6, the populations founded by old females that experienced low density had the highest fecundity. Similarly, when it comes to preference for finger millet, there was an increase from generation 1 to generation 6 in the young founder context, but the change was not significant in the old founder context. And at generation 6, populations founded by old females that experienced high density had the highest preference for finger millet. Given that founder context seems to influence oviposition choice of females in the 1st and the 6th generation, does this translate to changes in population size or differences in population size across the founder context? My expectation was based on the fact that females that have higher fecundity will give rise to populations that are large. On the x-axis in these populations is the generation, and on y-axis is the adult numbers. Individual replicate trajectories are shown in faint lines, and the bold lines represent the median trajectory for a given treatment. The red horizontal line represents a value of 100, which was the number of adults that initiated the population. at every generation. Contrary to our expectations, we do not see a significant difference among the population trajectories in the 4 founder contexts. But on average, populations founded by old females that experienced high density had the highest populations. I quantified population stability using a measure called the coefficient of variation. And I found that most of the populations had a high value of the coefficient of variation, indicating low stability. And this was regardless of, and this did not differ across the founder age and density context. So what have we seen so far? The age and density context of the founders influences oviposition preference in the subsequent generations. However, there is no significant impact of founder context on population size. So coming back to our question, does founder context impact colonization success? Potentially. And the impact seems to be through the influence of female fecundity and oviposition preference more than population dynamics. To continue this work, I am continuing experimental evolution to look at long-term effects on adaptation. I will also measure niche expansion in the adults, which gives us a measure of the percentage of finger millet consumed by the adults. And I would also like to investigate this question from a physiological perspective. I would like to look at whether the adults from the females from these different contexts differ in their anatomy. And I would also like to look at the females from these contexts as well as their eggs to see if they differ in their biochemical composition. With that, I would like to thank my advisor, Deepa Gashe, and everyone else who has been involved in this study. And I would like to thank you all for listening.
Great, thank you so much. And do we have any questions?
I can ask a question.
Absolutely, thank you.
Yeah, so this is an amazing talk, and so I've also been thinking about some of these concepts, but from an invasion ecology perspective, it's like what determines the colonization success of invaders? And I was like, it seems like a large part of context depends on like what is the context of the propagules that are introduced in a foreign territory. So have you ever like do you think about what do you say how this adaptation, or like how this context dependent might influence invasion success?
Probably, depending on what we see, it is also likely that invasion success can be influenced by founder context. And this will be more relevant because we are working with a pest species, and we want to see whether infestation could vary with context.
Good, thank you.